Very good afternoon, students. So, in the last class, we focused on regarding this TCA cycle PDH complex mechanism and its regulation, right? So, today let us see onto the next aspects of this TCA cycle, that is regulation of other regulatory enzymes of TCA. So, first of all, this regulation of this TCA cycle is like same that of the glycolysis, what we have seen in case of glycolytic pathway, which occurs at both the level of the entry of the substrates into the cycle as well as at the key reactions of the cycle. So the fuel which is going to enter the TCA cycle primarily as acetyl CoA, that is pyruvate to acetyl CoA formation, then the acetyl CoA which is going to enter. So the generation of this acetyl CoA from the carbohydrates is therefore a major control point of this particular TCA pathway. So this is the reaction which is going to be catalyzed by PDH complex, that is pyruvate dehydrogenase enzyme or multi-enzyme complex, which is called as. So we have discussed in the last class regarding this regulation as well as mechanism of that. So coming for this regulation of this PDH complex. So how you are going to regulate this? By allosteric modification and covalent modification of this in under this, and we can see this one is the first is the allosteric modification. So this PDH complex is going to be inhibited by acetyl CoA itself. That product itself is going to inhibit the regulatory enzyme of this first PDH or pyruvate dehydrogenase, and NADH, that NAD plus is acting as a cofactor, so it is involved in the synthesis of NADH. So NADH and activated by, so these acetyl CoA and NADH are going to act as a inhibitor, and it is going to be activated by non-acetylated form of coenzyme A, that is non-acetylated one. It is not acetylated one, non-acetylated form of this coenzyme A and NAD plus. So these two are going to act as an activator molecule for this PDH complex, that is pyruvate dehydrogenase complex. So what about this covalent modification? The pyruvate dehydrogenase are going to activate the pyruvate dehydrogenase activity of this particular PDH complex is mainly regulated by their rate of phosphorylation reaction. So we have discussed in their mechanism dephosphoenzyme and phosphoenzyme. Dephosphoenzyme will be active, phosphoenzyme will be inactive. So it is going to be regulated by their state of phosphorylation. So this modification is carried out by a specific kinase enzyme, as we have seen in the last, you might have gone through that slide, please go through if not. So PDH kinase will be activated by this acetyl CoA molecule in order to convert active form to the inactive form of this PDH. And the phosphates are going to be removed by a specific phosphatase enzyme, that is called as PDH phosphatase, right? So this kinase is going to be activated by NADH molecule and acetyl CoA, and it is going to be inhibited by pyruvate NAD plus and coenzyme pyruvate ADP, coenzyme A, calcium, and as well as NAD plus also, and magnesium, right? Calcium and magnesium in the muscle contraction. So this PDH phosphatase is, in contrast, it is going to be activated mainly by the magnesium ions, as I said you earlier while discussing this mechanism, right? So how this PDH complex is going to be regulated by allosteric modification and covalent modification. Coming for the regulation of other TCA cycle enzymes, so the most likely sites for these regulations are at the non-equilibrium reactions, so which are mainly catalyzed by citrate synthase, isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase, and these dehydrogenases are going to be activated by calcium ions, which in turn increase in the results in the increase in the concentration during muscle contraction and secretion. When there is increased energy demand, you can observe this particular activity of this dehydrogenase enzyme. So what is going to happen with respect to this TCA cycle enzyme? In this slide you can see, so the citrate synthase, that is the first enzyme of this, once after entering into the TCA cycle, that is once after entering into the mitochondria, once after the formation of this acetyl CoA, Now it is going to condense with the oxaloacetate to form citrate. So that was catalyzed by the enzyme citrate synthase. So there you can see make out allosteric inhibition of this citrate synthase mainly done by ATP molecule and long chain fatty acyl CoA. So this inhibition mainly done by ATP molecule and as well as long chain fatty acyl CoA and as well as isocitrate dehydrogenase is mainly allosterically stimulated by ADP molecule, which in turn enhance the enzyme's affinity for the substrates. In contrast to the NADH, in contrast to this, NADH is also going to inhibit isocitrate dehydrogenase by directly displacing NAD plus ATP. Those two is going to inhibit, showing the inhibitory activity, right? So it is going to directly displace the NAD plus and ATP 2 and it is acting as it is showing the inhibitory action. So this particular enzyme isocitrate dehydrogenase, you can make out it is mainly getting undergoing allosteric inhibition, right? Allosteric stimulation by the ADP molecule. ADP concentration is going to stimulate this activity of this enzyme, which enhances the enzyme's affinity for the substrate, whereas NADH is going to inhibit this enzyme by directly displacing this NAD plus and ATP concentration. High level of ATP is going to act as a inhibitor. Understanding? So coming for the next enzyme, there is alpha ketoglutarate dehydrogenase. This alpha ketoglutarate dehydrogenase is going to be inhibited by succinyl CoA and NADH, right? So the succinyl CoA and NADH molecule are going to act as a inhibitor of this alpha ketoglutarate dehydrogenase. In addition to this, you can also see this alpha ketoglutarate dehydrogenase is mainly inhibited by high energy change, right? So this high energy charge. So this high energy charge is also involved in the inhibition of this particular activity of the alpha ketoglutarate dehydrogenase. So the rate of the cycle is going to be reduced when the cell has a high level of ATP concentration. Of course, when the high level of ATP concentration, it is going to act as a inhibitor molecule and it is going to control the activity of this enzyme. So coming for the succinate dehydrogenase. So this is mainly inhibited by oxaloacetate molecule and the availability of the oxaloacetate is mainly controlled by malate dehydrogenase activity. So it mainly depends on the NADH and NAD plus ratio. So NADH to NAD plus ratio plays a very important role in controlling the activity of this succinate dehydrogenase. It is mainly inhibited by the availability of the oxaloacetate concentration, which in turn is controlled by malate dehydrogenase, mainly depends upon the NAD plus to NADH to NAD plus ratio concentration. So coming for the TCA cycle, other cellular ratio of this NAD plus, see how this NAD plus to NADH ratio is going to be maintained here. So in these three reactions which we have come across in the TCA cycle as well as PDH, which is going to utilize NAD plus as a cofactor, right? Three enzymatic reactions of TCA: isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase and malate dehydrogenase, as well as PDH, pyruvate dehydrogenase, which utilizes NAD plus as a cofactor. So the cellular ratio of this NAD plus to NADH, which has a major impact on the flux of this carbon through the TCA cycle. So the activity of this TCA cycle is going to be immediately dependent on the supply of this NAD plus concentration, which in turn, because of the tight coupling reaction which is going to happen in between oxidation and phosphorylation reaction, which is mainly dependent on the availability of the ADP concentration and hence ultimately on the rate of the utilization of the ATP molecule in the chemical and other physical work. Of course, it mainly depends upon the utilization. The rate of the reaction mainly depends upon the ultimately level of the ATP which is used in chemical and other physical work. So thus the respiratory control via the respiratory chain and oxidative phosphorylation, by coupling this oxidation and phosphorylation reaction, primarily regulates the citric acid cycle activity. In turn, we can regulate the citric acid cycle activity. So this NAD plus to NADH ratio plays a very important role in the flux of this carbon throughout this TCA cycle. So you can see here in the slide regulation of this TCA cycle, how the summary just I have shown you. Excess of the ATP, which is going to depict this energy rich state of the cell, hence TCA cycle is going to be inhibited, while reverse occurs when the cell is in low energy state with excess of ADP. So you can make out here which are all the concentration which is going to act here. See here, ATP molecule, acetyl-CoA and NADH are going to act as a inhibitor of this enzyme, that is pyruvate dehydrogenase. Here you can make out ATP and NADH same for the isocitrate dehydrogenase. ATP succinyl CoA and NADH concentration, which is going to act as an inhibitor for this alpha ketoglutarate dehydrogenase. So these are all the inhibitors molecule which you can come across in this particular TCA cycle. So coming for the significance of this TCA. So how it is important, how it plays a very important role in maintenance of this particular so this TCA cycle, how it plays a very important role and how it is going to act as a substrate molecule or any other intermediates of TCA which are involved in the synthesis of other molecules, right? So this citric acid cycle is not only a pathway for the oxidation of 2 carbon units, but is also a major pathway for the interconversion of metabolites arising from transamination reactions and as well as deaminations of amino acids, as we have discussed in the earlier. So transamination and deaminations of this reactions. So deaminations, example, glutamate to conversion of alpha ketoglutarate, that is complete removal of amino group, that is involvement of enzyme glutamate dehydrogenase with the formation of intermediate alpha aminoglutarate, whereas in case of transamination you can give the example formation of, that is amino acid plus keto acid gives rise to new amino acid and keto acid, that is transamination, transfer of amino group from glutamate to pyruvate to synthesize alanine, new amino acid to give you alpha ketoglutarate, that is a transamination. Transfer of amino group is happening from one amino acid to keto acid to form new amino acid and keto acid. So this also provides the substrates for the amino acid synthesis by transamination, right? These intermediates of TCA can also act as a precursor molecule or the substrate molecule for the synthesis of amino acids as by the transamination, as well as for the gluconeogenesis, as we have seen in the TCA cycle while discussing oxaloacetate you are going to come across, right? The formation of oxaloacetate, so that it is going to enter into the gluconeogenesis pathway and other fatty acid synthesis. So you can also come across with the acetyl-CoA molecule which can enter into the fatty acid synthesis involved in the ketone body formation and cholesterol biosynthesis mechanism, because it functions in both oxidative phosphorylation reactions, both oxidation and phosphorylation reactions. So this catabolic role of this TCA, first of all we will see the first importance or the significance of this TCA is catabolic role of TCA, particular intermediates of TCA. So the citric acid cycle is the final common pathway for the oxidation of this carbohydrate molecule, lipid molecule as well as protein, because glucose, fatty acids and most amino acids are metabolized to acetyl-CoA molecule or the intermediates of TCA cycle. So the functions of this citric acid cycle is the harvesting of high energy electrons from the carbon fuels, right? So that is one acetate unit generates approximately 12 molecules of ATP per turn of the cycle, as we have seen while discussing the energetics of TCA. So I have showed you how 12 ATP molecules are going to be the net ATP of TCA, right? So this is the catabolic role of TCA. So coming for the anabolic role of TCA, so as a major metabolic hub of the cell, the citric acid cycle also provides intermediates for the biosynthesis of various compounds. While discussing some anapleuritic and catapleuritic reactions, I have told you few just. So this role which is played by this particular intermediates of TCA in gluconeogenesis process, so all the intermediates of the cycle are potentially glucogenic, since they gave rise to the oxaloacetate formation, hence net production of the glucose, that is in the case of liver and kidney and other organs which are going to carry out this synthesis of glucose, that is through gluconeogenesis process. So you can see here anabolic role of TCA, how it is going to play role in this gluconeogenesis process. So you can make out the formation of oxaloacetate, that is by the enzyme pyruvate carboxylase. You can see formation of phosphoenol pyruvate carboxykinase, the formation of phosphoenol pyruvate, that is which are in this particular phosphoenol pyruvate is going to act as a intermediate for the synthesis or the precursor molecule for the synthesis of tyrosine, phenylalanine and tryptophan amino acids in plants and microorganisms, whereas in case of this gluconeogenesis process, phosphoenol pyruvate is going to act as a precursor for the synthesis of this glucose through gluconeogenesis process. And this phosphoenol pyruvate can also be utilized in serine, glycine and cysteine amino acid biosynthesis. So this phosphoenol pyruvate plays a very important role, anabolic role of this TCA just I showed you. So this is going to play a very important role in the plants and microorganisms with respect to synthesis of some amino acids. In case of you can make out through gluconeogenesis synthesis of glucose in human beings and other serine, cysteine and glycine amino acid synthesis also. So the key enzymes which catalyzes this net transfer of the out of the cycle into the gluconeogenesis is mainly done by this enzyme phosphoenol pyruvate carboxykinase, which involved in the catalyzation, which involves in the reaction of decarboxylation of this oxaloacetate to phosphoenol pyruvate with the GTP, which is going to act as a energy rich molecule, which is phosphate donor as well as phosphate donor in this particular reaction. Coming for the next role, that is in the synthesis of non-essential amino acids. It also plays a very important role, this TCA cycle intermediates, in the synthesis of non-essential amino acids. Since the transamination reactions which are going to be reversible, as you all know, so the cycle also serves as a source of carbon skeletons for the synthesis of some amino acids like alanine, aspartate, aspargine, glutamate, glutamine, etc. Right? So these are all the particular amino acids which are going to these particular intermediates of TC are going to act as a source of carbon skeleton for the synthesis of some amino acids, as I shown you here. So it is involved in the synthesis of glutamine as well as proline as well as arginine, right? So this through alpha ketoglutarate formation. So you will get in the transamination reaction, as I told you, glutamate which gives rise to alpha ketoglutarate, that is the transfer of amino group is going to happen from glutamate to pyruvate, pyruvate to alanine, alanine formation, and keto acid will be alpha keto. So this alpha ketoglutarate is involved in the synthesis of so many amino acids like glutamine, glutamic acids, proline, arginine, all those amino acids are going to be synthesized from this precursor, that is alpha ketoglutarate. In this slide you can make out, that is synthesis of non-essential amino acids. How the other intermediates of TCA are going to be involved in the synthesis of other amino acids, you can see here aspartic acid, which is going to act as a precursor of aspargine, lysine, methionine, threonine, isoleucine. So these amino acids are, except aspargine, are going to be the essential amino acids. They are synthesized only in case of the plants. So you can make out this aspartic acid which is involved in the synthesis of this aspargine, lysine, methionine amino acid synthesis. Aspartic acid is also involved in the synthesis of threonine. This threonine is also involved in the synthesis of so these are going to be acting as a precursor molecule, that is oxaloacetate for the synthesis of aspartic acid, threonine, isoleucine, and as well as pyrimidine nucleotides. So pyrimidine nucleotides can also be synthesized from this intermediate oxaloacetate. So coming for the role of TCA cycle intermediates in the fatty acid synthesis, so which is going to play a very important role, that's what I said you, acetyl-CoA, which is formed from pyruvate by the action of PDH, which is the major substrate for long chain fatty acid synthesis. And acetyl-CoA is going to be made available in the cytosol from citrate, which is going to be synthesized in the mitochondria, further transported to the cytosol, and it is going to be cleaved in a reaction catalyzed by ATP citrate lyase. So this how is going to happen in case of fatty acid synthesis. This intermediate play a role in fatty acid synthesis. Coming for the other particular molecule, that is a citrate, how it is going to be transported out of the mitochondria when aconitase is going to be saturated with its substrate molecule. So this is going to ensure that citrate is used for the fatty acid synthesis only when there is an adequate amount of the ensured continued activity of this cycle is going to happen. That is acetyl-CoA can also used in the synthesis of other cholesterols and steroid molecules, you can see here. So you can make out citrate, that is through in the cytoplasm you can see ATP citrate lyase to form, it is going to cleave this molecule to form oxaloacetate plus acetyl-CoA. Then this acetyl-CoA is involved in the synthesis of steroids, fatty acids and other cholesterol that is involved in the cholesterol biosynthetic pathway. Coming for the role of this TCA cycle intermediates in the biosynthesis or heme synthesis. So succinyl CoA, very importantly, which plays a very important role, the succinyl CoA majorly. So this succinyl CoA, which is going to condense with amino acid glycine to form alpha amino beta keto adipic acid. You can make out here alpha amino beta keto adipate, which is going to be formed, which is catalyzed by this ALA synthase enzyme. Then delta amino levulinate is going to be formed. This is the synthesis of perfobilinogen or the synthesis of heme. These are the reaction which is going to, after that, two molecules of delta amino levulinate is going to condense, catalyzed by ALA deatatase, which is a zinc based enzyme. So you can see perfobilinogen synthesis, perfobilinogen, which is a very important molecule for the synthesis of the heme. So this is the first step, formation of alpha amino beta keto adipate in the heme biosynthesis. You can see how it is going to play a role. Just I have shown you in this slide. So how this succinyl CoA, which is going to involve in the synthesis of heme by condensing with amino acid glycine. Coming for the role of other intermediates of TCA in purine and pyrimidine synthesis, you can make out glutamate and aspartate, which is mainly derived from this TCA cycle, are utilized for the synthesis of purine and pyrimidine. That's how citrate molecule in fatty acids, steroids, and alpha ketoglutarate, glutamate, and other amino acids to purine. You can make out here porphyrin, heme, chlorophyll. In case of succinyl CoA, oxaloacetate, it results in the formation of aspartate, and then further other amino acids and purine and pyrimidine can also be synthesized. Coming for the role of vitamin, this is the last slide. So you can see here riboflavin, which is the form of the flavin-adenine, it is going to exist in the form of FAD, that is flavin-adenine dinucleotide, which is acting as a cofactor for the enzyme, especially in the TCA cycle. Very importantly, this enzyme is very specific for this cofactor, that is FAD molecule, that is succinate dehydrogenase. So the niacin, which is in the form of nicotinamide adenine dinucleotide, that is NAD+, the electron acceptor for this isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase and malate dehydrogenase is the NAD+, except succinate dehydrogenase, which utilizes FAD as the cofactor. Because it cannot transfer directly from NAD+ electrons, so FAD is going to act as a cofactor there. Thiamine, which is going to act as a coenzyme, vitamin B1, thiamine TPP, thiamine pyrophosphate, which is the coenzyme for the decarboxylation of this alpha ketoglutarate dehydrogenase reaction to carry out, and pantothenic acid, which is acting as a part of this coenzyme A, such as acetyl CoA and succinyl CoA and oxaloacetate, which in those reactions involving those molecules with respect to this reaction, which plays a very important role in this particular acting as a vitamin, which plays a very important role in the synthesis of this vitamins as a coenzymes are the cofactors. So these intermediates are involved, these cofactors are involved in the TCA cycle. So this completes your TCA cycle, complete regulation, steps, mechanisms, significance, everything. If you have any doubts, please go through. This is very important and you can talk to me later. Thank you.